Hello and welcome from the Royal Swedish Academy of Sciences, where we've just discovered who has received the 2009 Nobel Prize in Chemistry. Joining me to discuss this prize is Professor Gunnar von Heine, the chairman of the Prize Committee in Chemistry. Professor von Heine, thank you very much for joining us straight after the announcement. Could we begin by you revealing who has been awarded the 2009 Nobel Prize in Chemistry?
So the three laureates are Venki Ramakrishnan from the UK, Tom Steitz from Yale University in the States, and Ada Yonath from the Weizmann Institute of Science in Israel.
And what have they received the prize for?
So they received the prize for their studies on the structure and function of the ribosome.
And to anyone who's not familiar with what a ribosome is, can you explain why it's important and why it's important to know its structure and function?
Yeah. So our bodies are, the molecules that make our bodies work are the proteins. Somehow the body has to make proteins, and the blueprints for the proteins are stored in the genetic material, in DNA. Then when a particular protein is to be made, a little piece of DNA is copied into something called a messenger RNA, and finally a ribosome, and now we get to the prize. A ribosome reads off this mRNA molecule, and according to the instructions carried by the mRNA molecule, the ribosome puts strings together amino acids into a specific sequence that will then make that particular protein. So ribosomes are absolutely essential to the life of the cell, and it's the only way that cells can make proteins.
And so by sort of viewing its structure and understanding its function, what can we hope to learn?
So first of all, of course, it's a deep scientific question, is how does the ribosome work? This very complex machine, it's a big molecular machine, and it has very intricate workings. And of course there is this urge on the part of the scientists is to understand how these machines work, like the ribosome. Of course there are also more applied aspects of this knowledge which has been gained by these studies, and in the case of the ribosome in particular it deals with antibiotics and antibiotic resistance and the possibility to develop new antibiotics.
And we'll talk a bit about the antibiotics and antibiotic resistance a bit later, but could you tell us a bit about what tools and techniques the three laureates used to uncover the structure and function of the ribosome?
So they mainly used a technique called X-ray crystallography, which is a way to determine with very high precision and resolution what molecules look like. And the ribosome, after all, is a molecule. It's an extremely complex one. So it's a very difficult problem to solve the structure, to understand how the atoms are arranged in the ribosome. But these three laureates have actually solved that problem. Now, X-ray crystallography, what you do is you purify ribosomes, which in itself is an accomplishment. Then you have to coax these ribosomes to form crystals, like a salt crystal, but a ribosomal crystal is much more difficult to make than a salt crystal. And finally, you take your ribosome crystal and you shine X-rays onto it. These X-rays will scatter from the ribosomes in the crystal, and from the scattering pattern you can, if you're good enough and lucky enough, you can calculate what the actual structure of the ribosome is.
And so how difficult a task was it to be able to get X-ray images of ribosomes?
So this was a very difficult task. It took more than 20 years. The first problem was, as I said, was to be able to even to make crystals. And Ada Yonath is the pioneer in this field. She made the first, for a long time she was alone, and she made the first useful crystals of ribosomes, after which Tom Steitz and Venki Ramakrishnan came in and also in parallel with Ada Yonath started to actually solve the structures of the ribosome.
And so once the structure of the ribosome had been identified, then scientists tried to look at how it functions. And this also involves the work of the three laureates. Could you discuss that more, please?
Yes, so from these structures that the three laureates and a few other people, but mainly the three laureates, have produced, we have now learned how the ribosome can be so precise in selecting amino acids to be strung together into proteins. So it's a problem of accuracy. How can it be so accurate? And we have also learned how the actual stringing together works. How does it work from a chemical point of view when one amino acid is tagged on to a growing chain of amino acids to make the protein?
And what were the key discoveries involved in understanding that function?
So both these discoveries were again based on very accurate structures of the ribosome in various functional states. So it's not enough just to take one ribosome and make one crystal and you get the whole answer, but you have to make very different preparations with ribosomes bound to other small molecules or other molecules that take part in the process. And only by studying these complexes can you then hope to learn something more about the function. And this is precisely what these laureates have done over the past 5 or 10 years.
And specifically, what were the achievements that the laureates made in terms of understanding this?
So what they have shown us is, number one, how can the ribosome be so accurate? So we now understand this on a molecular level, how this works. Number two, we understand how the actual synthesis of the protein is carried out by the ribosome. And we also understand actually a few other aspects of the whole process, which which also have come out of this work, but maybe going too far to start describing these in detail.
And so that understanding the structure and function tells us about how the ribosomes create the proteins that sustain life, but also it's given us insights on how to save lives with the way that antibiotics work. Could you discuss a little bit about how antibiotics function and how the achievements from the laureates have helped us to create better treatments?
Yes. So antibiotics, of course, are small molecules generally that disturb one or other function of a bacterial cell and kills the bacterium. It turns out that about half of all the known antibiotics actually have the ribosome, the bacterial ribosome, as a target. So they stop it dead in its tracks, basically. Up to the point when we started to get these structures from the laureates, nobody really understood how these antibiotics could damage the ribosome, the bacterial ribosome. With their work, number one, we understand how these different antibiotics work, why are they toxic to the bacterium, but moreover, we also now have a possibility to take this knowledge and use it to design new antibiotics that may help us conquer or at least push forward the resistance problem. Bacteria develop resistance to various antibiotics.
So if I can sort of move towards the prize itself, as you implied, this research has taken decades and it's answered some questions. There's still some questions to answer, of course. When deciding that the prize should go to these people, was there any recent developments or recent discoveries that made it a more compelling case to select this prize this year?
So the main breakthrough here from the three laureates was in the year 2000-2001, when the first really good structures were published. But in the interim, mostly these three laureates, but also some other people, have been able to make these different complexes that have illuminated various aspects of the function of the ribosome. So they have continued to contribute up till the present time, actually, and they still are. But there is a number of very important steps in the interim that have also contributed here, and that were absolutely essential for us to be able to award this prize.
And one thing you mentioned briefly earlier was the contributions of other people to the field, and sometimes when you're selecting a prize, the laureates are very easy to pick, and sometimes they're a bit more difficult because there's so many key players in the field. What was it like this time? Was it an easy, straightforward decision to make, or did it require discussion?
Well, it's never easy. I can tell you that. It's never easy, even if it seems like it. But so clearly this prize is a prize that has been discussed for years in the committee, and we've been through every possible aspect of it, we think. And in the end, we come to a decision. It's never an easy decision, but we make a decision.
And so the criteria for the selection of these, could you sort of summarize why you think these were the key achievements in understanding the structure and function of the ribosome?
Well, to go back to what I said before, these are the three key people who have made the determination of ribosome structures possible, and they have also pushed the analysis such that they have uncovered these very important functional aspects of how the ribosome actually works. So it's from this point of view, it was a fairly straightforward decision.
And have you spoken to the laureates at all?
Yes, we have. We call them, of course, as we always do, and we managed to reach all three of them. Well, it's always interesting to have these conversations with people who are woken up in early morning hours. One was Thomas Stites was on his way to the gym, and I think he had to forget about the gym for this today, but
just for one day perhaps. Yeah. And it's worth noting, of course, that Ada Yonath becomes the fourth woman laureate in chemistry, which is a nice achievement, isn't it?
Yes, it's 45 years since there was a woman won the chemistry prize before. So,
well, Professor von Heene, thank you very much for discussing this with us. For more information on the Nobel Prize in Chemistry, please visit nobelprize.org, where you can see the announcements and will soon be able to see exclusive interviews with the new laureates. That's it for now for this live webcast, but join us tomorrow where we'll discover who will be awarded the 2009 Nobel Prize in Literature. The announcement's expected at 1 p.m. Central European Time. But for now, it's goodbye from all of us at nobelprize.org.